Hiya, this is just a quick tutorial on how to use ImageJ and how to start analyzing your pictures. So hopefully you've been able to download Fiji. Fiji is just ImageJ, as you can see here. And if you want to open the pictures, just open this CZI format. Should be fine. Usually here you don't have to change anything. You can just press OK, and then hopefully you'll get the images looking like this. And you can see here's the purple channel, here's red, here's green, and here's blue. But the problem is that they're all separated. So what you need to do is that you need to actually separate them again. You go to image and then color and then split channels, and then they're going to be separated like this. And then you need to merge them again. So go back to color, merge, and then you can decide here what's going to be red, green, blue, and gray. And usually it's fine just to leave it. And then create a composite. If you make sure this is clicked, create a composite, and then you get them all aligned like this. You can now zoom in by pressing Control and scrolling on your mouse. But that's the problem. As you can see, we're seeing all the images all the colors at the same time. So what we need to do is open the color channel tool. And if we open the channel tool, we can turn off the blue, which in this case is EDU, and we can turn off the channels that we're not really interested in, and we can go back and forth. So if we look at this cell here, it's clearly EDU positive, and then we can turn off the EDU, see that it's also positive for HopX. Doesn't seem to be positive for KL67. Unluckily, we have a DAPI staining there, so it's a nucleus, and that's what we'd expect. So that's the way to count the cells here. You can systematically go through it. There are also plugins you can use. For example, Cell Counter is a plugin where the software can count the cells for you. It can be a bit tricky to get it started, but once you get it started, it should be faster. You can also adjust the brightness and contrast, and you need to decide which color you're adjusting. You need to move this bar here. So here now we're adjusting the purple. We can reset. Then if we move it here, now it's the red channel, now it's the green channel, now it's the blue channel. So maybe we want to just, so this is the green. This staining didn't work that great, but we can adjust it. We can also try and see what outer looks like, but that basically looks quite messy. So probably it's just best to leave it like this. But here's where you can adjust that and you can do that one at a time. And then if you want to export these to put in your report or whatever you want to do with your images, you can push save as and then JPEG or TIFF, and then you can just save them as whatever file format you want to save them as. And that's about it for this first video, and just learning how to open images and do some easy counting in ImageJ. And remember that if you do any adjustments here, you need to make sure that you do the same adjustments on the control and the treated group. You need to be, the best thing is if the pictures, if you don't need to do any adjustments. Thank you very much.